Hi, everyone. My name is Camryn Pettenger-Willey, and I am an intern at the Pacific Northwest National Laboratory this summer, which is in Washington. And I am also an undergraduate at Whitman College, and I am majoring in biology with a minor in computer science and in chemistry. So for my internship, I actually ended up working on two projects. So the first project is the protocol optimization for enzymatic digestion with mass spectrometry imaging. And the second one is multi-image alignment and resolution improvement. So first, to just go over a quick agenda of what I'll be going over. I'll talk first about the protocol optimization project, which was more experimental wet lab. And I'll go over the background, the process that we went through, and then some of the results that we ended up with. And then I'll be talking about the image improvement project, which was more computational coding. And I'll introduce the goal, the processing and alignment steps we had to go through, and then the next step that we still have to take. So first off, to give a little bit of background on mass spectrometry. Mass spectrometry is a type of analytical technique that is used to measure molecules based on their mass to charge ratio. And most instruments usually consist of three main parts. There's the ionization source, the mass analyzer, and the ion detector. And you can see over here on the left is a simple diagram of what most of them look like. And then also on the right side, there are two of the instruments at our lab. There's the UHMR and then the CIMAX. And we collected most of our data on the CIMAX. Now, to talk a little bit more specifically about MALDI imaging, MALDI imaging is a type of mass spectrometry that just differs in the ionization source. So MALDI stands for matrix-assisted laser desorption ionization, which basically just means that there's a laser that is fired at the surface of the tissue that helps to break off small pieces of the sample and allow them to be analyzed. MALDI imaging also uses a matrix to help absorb and transfer energy. And it's a pretty cool technique because it allows you to create these spatially mapped images of biological molecules such as proteins, metabolites, lipids, and more. And you can see that in this mouse brain image here. Now, our project wanted to specifically focus on proteoforms, which means we're interested in how the proteins are being changed or modified after they are formed with these post-translational modifications. And there are two most common techniques that are used to analyze proteins. There's bottom-up, which means taking the whole protein and then breaking it into really small pieces for analyzation. And then there's the top-down approach, which takes in the whole intact protein molecule. However, both of these approaches do have their downsides, and current instrumentation really struggles with the large masses for the top-down approach. So because of this, we wanted to introduce a middle-down technique using glucose digestion. And this allows us to create medium-sized fragments that helps complement top-down to create a more comprehensive analysis of these post-translational modifications. So it was important that we modify this protocol for this glucose digestion because it allows us to improve the resolution of these combinatorial PTMs because of the size of these medium-sized fragments, smaller than top-down. And so this also works well for really highly complex proteoforms, such as histones. So in order to go throughout this process, there's three main steps. First, you have to do the tissue preparation, which involves slicing, washes, sprays, and then to actually collect the data, you have to do MALDI imaging. And then in order to assign the peptide masses, there's a bunch of data processing that you have to go through. So our goal was to optimize the tissue preparation protocol in order to improve the results that we were seeing. So this slide is a bit of an overview of the many steps that it takes to prepare our tissue. And I'm just going to highlight a couple here that we ended up modifying and we saw improved results with. And here are some images that we ended up with. You can see there's the comparison of 2-hour incubation versus 24-hour incubation. And this is one of the final steps that we took throughout our optimizing the protocol. And you can see that the 2-hour incubation images have much clearer structures and you can really see a better signal intensity. So this is what we're aiming for, and these are results that we were happy with in the end. And you can also see the data at the bottom comparing the 2-hour results in pink. There's much better signal intensity peaks there. So this is a bit of an overview of the changes that we all made to improve the results. And we made all these changes in order to change this image that we started off here in order to create much clearer images with better signal intensities. So for example, we ended up decreasing the length of incubation time to 2 hours because this allowed us to see less delocalization of the peptides in our sample. And this allows us to create clear images, which is the goal. And finally, the next step for this process is to actually assign the proteins and do this peptide identification. And now I'll be talking about my other project that I worked on, which is the image alignment and resolution improvement. And this was all done through coding in Python with the OpenCV and Pillow libraries. So our goal for this project was to improve the resolution of those MALDI images through combination with histology images, which typically have a much better resolution. And MALDI images are usually somewhere around 100 microns, and this is based on time efficiency and instrument limitations. So in order to go through this whole process, there's many steps that must be taken, such as cropping the images, maybe filling in holes in the tissue, and also lining up the two images on the same coordinate system. So these are some of the tools that I created, and here are some example images of the examples that I did. So here is the mouse brain tissue and kidney tissue, and you can see that the outside is cropped out and made transparent. And for these, I use functions such as thresholding, morphological transformations, and masking. And here are some of the histology tissue images. These ones also, you can see the background is cropped out, and then there's also some holes filled in the middle that are made transparent. Now, the next step for this process is the coordinate matching. So you have to take the two images and find features that line up and input these coordinates in order to warp one image to the other to get them to perfectly align. So once this is done, then you can create an overlay of the images. And I also created this plot to compare the three images. There's the MALDI, histology, and then the overlay of the two. And it has synchronized zooming and panning between the three. So you can double check how you align them and maybe change some coordinates if it doesn't look exactly the way you want it to. And finally, the next step for this project will be predicting the finer resolution. We need to sharpen the resolution of the MALDI images based on a regression model to predict the finer ion distributions. And I still have a little bit of time left in my internship, so that's what I'll be working on then.
2 minutes remaining.
And just to talk a little bit about my internship experience, I have honestly learned so much about mass spectrometry and gaining a lot more confidence in the lab. And also coming into this internship, I barely had any experience in Python before, but now I know how to make my own functions and use a lot of tools with image processing. And then also as a side note, we have 2 manuscripts in preparation, so that's pretty cool. And now to give some thanks to everyone, because this could not have been possible with everyone that helped me along the way. So a big thank you to the National Institutes of Health and HuBMAP for giving me this opportunity and funding my internship. And thank you to the Pacific Northwest National Laboratory and especially my team at PNNL. Thank you so much. I've honestly learned so much throughout this internship, and they've really done a great job of mentoring me. Thank you.